The last thing we're going to talk about in today's lecture is the concept of water availability. This concept tells us that the literal amount of water that's in the environment around a microbe is not what's important. What's important is how much of that water is available for the microbe to use. Now, we've talked in here before about the fact that microbial cells are very susceptible to the effects of osmosis. I have a reminder down here on the slide of the fact that osmosis can cause water to rush into a cell or rush out of a cell, and it can actually have deadly consequences to a cell. So changing concentrations of dissolved molecules around a cell really matters, because if those molecules are unable to cross the membrane and a concentration gradient is created that cannot be resolved, osmosis will result, and that can have devastating effects on a cell. Remember, the cell wall is there around a bacterial cell, and it does provide some protection from osmotic shifts. And that's good, because otherwise a lot of bacterial cells would experience osmosis as their environment changed. And again, that osmosis could end up destroying the cell. Water is absolutely required for all microbial cells, for all cells, because life is water-based on our planet. Living things contain 70% or so of water. Water is essential. Water is required for many, many, many metabolic reactions. But that water can also be problematic for a cell. Now, when it comes to water availability, we're talking about something very different. Water activity is the measure of water that is available to microbial organisms in a particular environment. Pure water, water that has no other substances dissolved in it, has a water activity of 1. Now, we shorthand water activity with a lowercase a and a subscript W. Pure water has a water activity of 1. All other water activity values fall between 0 and 1. And the lower the water activity, the less available the water is to microbes in that environment. The reason we care about water activity and the reason we measure it in certain foods is because it gives us an idea of how likely that food is to get contaminated with microbes. We know the water activity for a lot of different types of food, and it gives us information that tells us how we need to store that food or keep that food in order to prevent harm to people who eat that food. There are a lot of foods that have a low enough water activity that they can be kept on the shelf, for example, and not refrigerated. You don't have to cook them before you eat them, because the water activity is so low that the chance of microbes growing in that food is very low. We've used a lot of different methods to preserve foods throughout the centuries of human existence. We've used everything from drying out certain foods to salting certain foods, and even using sugaring as a method to prevent bacterial growth. In modern day, we know a lot about how to keep food safe. And part of it is how we preserve the food, and part of it is just how likely it is for anything to grow in that food. On the topic of sugar, you may or may not be aware that foods that have a high sugar content, especially foods that are sort of syrupy or thick, those foods have a very low water activity. And it's extremely hard for bacteria particularly to grow in those foods. Sweet things have a lot of sugar, a lot of carbohydrate. And the way carbohydrate interacts with water is going to reduce how much of that water is available to the microbe. If we go back to this slide for a minute, and this diagram over here on the right, you're looking at a sodium ion here, and it's surrounded by water molecules. Remember, water is a polar molecule. And as we've discussed in here, polar molecules have partial charges within them. The oxygen atom has a higher electronegativity than the two hydrogen atoms. That means that the electrons in the covalent bonds are pulled more closely to the oxygen than they are to the hydrogen. And that creates partial charges. The oxygen is partially negative, the hydrogen is partially positive. Now the sodium ion, it's not partially charged, it's fully charged. It has a positive charge. It's an ion. It's a cation, a positively charged ion. That positive charge is going to attract the partially negative oxygen atoms in water. And what's going to happen is water will surround this ion. These are weak attractions, but they're attractions. And this sodium ion is going to get what we call hydrated. It's going to be surrounded by water molecules. And there'll be weak attractions between the partially negative oxygen and the positive ion. That makes the ion less available. It also makes the water less available because those two things are sort of bound up with each other. So when it comes to sugar, sugar or carbohydrate is also polar. And so it's going to contain partial charges, and it too will be attracted to water. Water and sugar will hydrogen bond with each other. Carbohydrates get hydrated in the environment around a microbial cell. That means that that water is less available to the microbes. It's interesting because fungi are not nearly as affected by water activity as bacteria are. Probably because fungi tend to grow on the surface of foods, not in the interior the way bacteria tend to grow. A good example of this sugar effect can be seen in foods like honey and foods like maple syrup, even jams and jellies. If you buy a jar of jelly at the grocery store and you use it and you keep it in your fridge, you've probably noticed that as the jelly gets older and older, there may come a time when you open up that jar and you can see some fungus growing on the surface. You can see some mold growing on the surface of the jelly. Again, fungi are not nearly as affected by water activity as bacteria are. That jelly can get infected, or I should say contaminated, with fungi, but it's unlikely to become contaminated with bacteria. Even very old jars of jam or jelly, very unlikely to get contaminated with bacteria, and that's because the water availability is too low in that thick, sweet food. Honey is another example of a very sweet food that is sort of thick and syrupy. In ancient Egypt, honey used to be used as an embalming medium. The dead body would be embalmed with honey because, of course, human bodies are covered with microbes and full of microbes, and the honey would prevent those microbes from growing and help preserve the body. You can keep a jar of honey for a long, long time, as long as you prevent fungi from getting in, because again, they can grow on the surface of the honey. As long as you prevent that, bacteria just won't grow in honey. Maple syrup is another good example. While sometimes fungi can grow on the surface of maple syrup, you just don't get bacteria growing in that food. And that's because it's just full of sugar. It's full of carbohydrate. Thick and syrupy. If we go back to this slide for a second, what happens with those sugary foods is the individual molecules of carbohydrate, the individual molecules of sugar would be sitting right where the sodium is. They end up being surrounded or hydrated with water because the sugar has partial charges, the water has partial charges, and the partially negative and partially positive areas are attracted to each other. That makes the water less available to the microbe. In many textbooks you can find charts like this one. This chart shows us the water activity in several different food substances. You can see again pure water at the top. Remember, pure water, which has nothing dissolved in it, it has a water activity of 1. All the other foods listed here, or substances listed here, are between 0 and 1. Notice human blood, which is primarily water, has a water activity of 0.995. If you travel down, you'll see seawater under here has a slightly lower water activity. Bread. Then we go down to maple syrup, ham. Remember, ham is not full of sugar. Ham is full of salt. Same idea with the salt. Salami is next. Fruitcakes, jams, I mentioned. Salted lakes, salt fish. And finally, really dry foods like cereals, candies, dried fruit. They have the lowest water activity. So by looking at these water activity values and remembering that it's harder for microbes to grow where there's a low water activity, it's easier for bacteria to grow in human blood than it is for them to grow in, say, maple syrup. It's easier for them to grow in maple syrup than it is, say, for them to grow in dried cereal. So you can use this information to help you know how to handle food safely. And this is where a lot of our food safety recommendations come from. You don't have to keep a box of dried cereal in the refrigerator because the water activity level is so low, it's unlikely that bacteria will grow in it at all. In comparison, even though maple syrup has a lower water activity than, say, water, we still want maple syrup in the fridge. After you open it, you should keep it in the fridge. It's not likely that bacteria will grow in it, but we want to prevent any bacterial growth as much as we can. So, high sugar foods, high salt foods, very dry foods are foods that bacteria just have a difficult time growing in. Remember, we used specific language when we talked about an organism's ability to tolerate heat. We talked about mesophiles and thermophiles and so on. We also have specific language when it comes to their ability to tolerate salt. We talk about halo-tolerant microbes. Halo translates as salt. So an organism that is halo-tolerant is an organism that can survive in foods or substances that contain between 0 to 5% sodium chloride. Remember, blood, like all human fluids, contains 0.9% sodium chloride. That falls well within this range, and there are lots and lots of different organisms that can get into our blood and grow. We give that problem a name. We call that sepsis, when bacteria get into the blood and grow. Blood has salt in it, but it's a low enough amount of salt that many microbes are able to tolerate it. There are some microbes that are halophiles. They love salt. So they can grow in environments that have anywhere from 5 to 20% salt. That's a lot of salt. Most of the halophiles are actually archaea. Remember, the archaea in general can survive in more extreme environments than bacteria can. There are even extreme halophiles These are all archaea. They can survive in concentrations between 20 and 30% salt. That's a lot of salt. For comparison, the salt in ocean water, in marine water, is about 3 to 5%. So that's an awful lot of salt. And there are some microbes that can grow in that amount of salt. Lucky for us, lucky for humans, remember the archaea don't cause disease in us. So even if they do grow in, say, salted foods, foods that have been salted in order to preserve them, even if archaea can grow in those foods, we don't develop disease if we consume those archaea. There is one particular pathogen I want to mention here, and that's Staphylococcus aureus. Staphylococcus aureus is a problematic pathogen because it can survive in the types of salty foods that would kill most other bacteria. One of the biggest problematic foods that gets contaminated with Staph aureus is ham. If you go to the CDC website and you investigate instances of food poisoning, ham is one of the big players that shows up again and again in food poisoning outbreaks. What happens a lot of times is ham gets brought to a function where people are eating, and the ham will be sliced up and left out at room temperature for long periods of time. Now, most bacteria can't grow in ham because it has too much salt in it. But Staph aureus doesn't mind that salt. It can tolerate that salt very well. And so again, when you combine the problem of this particular meat, which tends to grow bacteria anyway, and then you leave it out at room temperature for long periods of time and allow people to come up and take some and bring it back and eat it, that can cause outbreaks of Staph aureus food poisoning.